Understanding cancer is the fundamental point of research, really. We need to understand the molecular basis of cancer so that we can develop targeted drugs to those cancers. I'm Professor Stuart Pitson here at the Centre for Cancer Biology in Adelaide. I head up the Molecular Therapeutics Lab that's developing new therapeutics for acute myeloid leukemia. Acute myeloid leukemia is a very common blood cancer. It's essentially where the production of blood cells is defective, and so the bone marrow where blood cells are normally produced gets clogged up with ineffective cells, and so the patient can no longer develop normal blood cells, and so they become anemic and often die. The problem we're trying to solve really is that acute myeloid leukemia is not very well treated at the moment. So generally patients receive standard chemotherapeutics which are pretty toxic and not very effective at being able to cure disease. So what we're trying to do is develop new targeted therapies that can eradicate the disease and give an actual cure. Progress has been good. We've identified a metabolic vulnerability in this disease. We've also developed a drug that can target that metabolic vulnerability, and it shows great effect in being able to kill acute myeloid leukemia cells and leave normal cells untouched. We've found that our approach actually seems to work with in combination with a number of different drugs. So it's pretty exciting, I guess, that we might have something that could work across a range of different drugs and a range of different subtypes of this disease. The impact means that we may be able to actually treat the disease better. So at the moment, patients will receive a cocktail of different drugs. They often respond initially, but then they relapse. And so the idea is that we can combine our drug, which is still in development, but our drug with this different cocktail and then actually eradicate the disease fully, so that those patients then will not relapse and can live long and healthy lives after that. The modern technology we use really is genomic sequencing. That's advanced enormously in the last 10 years. It helps us to understand the disease much better and to be able to target more selectively acute myeloid leukemia cells. The problem with that, though, is it's incredibly expensive. Each patient sample we would have would cost around $1,000 per sample to analyse, and so we may analyse 50 to 100 samples a year. The future of cancer research really, I think, is centred around personalised medicine, so understanding the mutations that occur in cancer and being able to target those selectively with drugs. So the CCB is a medical research institute here in Adelaide. The building we're in at the moment is the Bradley building. The top 4 floors are the Centre for Cancer Biology. There's around 20 labs. We're in the wet lab here of my lab. This funding is part of a much larger program that we've actually been operating for about 20 years. So this is the culmination of 20 years of work that we've been undertaking. In the last 20 years, we've understood a lot of fundamental biology around acute myeloid leukemia. We've identified molecular targets. We've developed small molecule drugs that can be potentially used. RALLIES funding is really then allowing us to use that, put all those things together, use that drug-like molecule and be able to push that towards a clinical trial. And that's really our goal, is to get these drug-like molecules into patients and help patients.